Good afternoon, everyone, and welcome to our 17th edition of our INFOGEST Viva webinar. My name is Hernandez Olivas, and I would like to introduce Cynthia Fontes, who will conduct the webinar with me. This event is organized by a team of postdoc from Teagasc Food Research Center in Ireland. This webinar aims to share the latest findings in digestion with presentation from the most recently graduated PhDs, postdocs, and young researchers. This webinar is broadcasted live on YouTube and will be accessible on the INFOGEST YouTube channel. The presentation today in a pre-recorded video will be followed by a question and answer session with the speaker. So I actively invite you to use the Q&A button. It should be around here. Our speaker today is Maria Vivanco Maroto with anorexigenic hormone release in response to protein digestion products. She currently works in the food proteins group in the Food Science Research Institute, which is a mixed research center belonging to the Spanish National Research Council and the Autonomous University of Madrid in Madrid, Spain. She has recently defended her thesis in which she had studied the secretion of anorexigenic hormones in response to peptides release during gastrointestinal digestion of food products. During this period, Maria has been able to do research stays in both nationals, like in Spain, in the Universidad Rey Juan Carlos and IMDEA Alimentación, and also international stays in the University of Lille in France. So, Maria, the floor is yours.
Hi everyone. Today I'm going to present a part of my doctoral thesis where I studied the anorexigenic hormone release in response to protein digestion products. This thesis was supervised by Isidra Recio and Beatriz Miralles. The gastrointestinal digestion of food proteins results in about 30% amino acids and 70% peptides, depending on the protein susceptibility to this process. After digestion, the resulting products make contact with brass border enzymes that continue the hydrolysis process. And finally, these products could exert local effects or they may be absorbed. The gastrointestinal tract has different cell populations. Enteroendocrine cells are just 1%, but represent one of the major endocrine organs. In response to protein digestion products, these cells release various anorexigenic hormones like CCK, PYY, and GLP-1. In addition, signals released by enteroendocrine cells can regulate insulin secretion by the pancreas or control energy homeostasis. This image shows the protein-sensitive receptors and their location along the gastrointestinal tract. In blue, you can see that the GPR93 receptor is the most highly expressed in this tract. And in addition, it's important to say that this receptor is co-leveled with the cells that release PYY and GLP-1. Protein digestion products induce the release of CCK and GLP-1. CCK stimulates the secretion of pancreatic enzymes and bile, promotes insulin secretion, inhibits gastric emptying, induces satiety, and increases intestinal contractions. On the other hand, GLP-1 enhances insulin secretion, suppresses glucagon secretion, lowers glucose levels, inhibits gastric emptying, and induces satiety. For that, in the last years, GLP-1 analogs have been developed in order to treat pathologies like obesity or type 2 diabetes. Peptides and amino acids can release these hormones, but the structural requirements necessary to induce their secretion are not totally known. For that, the main objective of this thesis was to evaluate the hormonal response to dietary protein digestion products, especially milk and egg proteins, in relation to the release of anorexigenic hormones CCK and GLP-1. The first objective was to study the digestion process of milk caseins and whey proteins through gastrointestinal simulations and to analyze the metabolic response in relation to the peptide and amino acid profile. The second objective was to identify peptides released during gastrointestinal digestion that induce the secretion of anorexigenic hormones CCK and GLP-1 and to understand the receptors involved in this process. First, we evaluate the inclusion of brass border enzymes in the intestinal phase of the INFOGEST protocol. For this, we used casein and whey as substrates and perform digestions following the INFOGEST protocol. Also, we followed this protocol and we add BBM in the intestinal phase. And after, we characterized the amino acid and peptide fraction by HPLC-MSMS, and in addition, we compare the peptide sequences with those previously found in human jejunal aspirates, and also we study the in vitro bioavailability of resistant sequences. These graphs shows the percentage of peptides generated during the digestion process following the INFOGEST protocol in blue, with the addition of a low concentration of BBMs in orange and a higher concentration of these BBMs in gray. For both substrates, the data show that adding BBM in the intestinal phase of the INFOGEST protocol increased the number of peptides generated. For casein, there is an increase in peptides between 5 and 9 amino acids, but in the case of the whey, this increase is in peptides with 5 amino acids. For that, these results indicate that the addition of BBM continues the protein hydrolysis process. This table represents the peptides generated in the control digestion and those generated by adding BBM. The data show that the main activity of these BBMs is the aminopeptidase activity, but also we look at carboxypeptidase activity of these enzymes. This image represents the probability that a specific amino acid will be in the first, second, or third positions of the amino or carboxy end of the peptide sequence. On the left, this probability is shown for all in vitro conditions, and on the right, we showed the profile previously found in human jejunum. For casein, the probability of proline appearing in these positions is common in all conditions, but in the case of the whey, the presence of glutamic acid and aspartic acid is common. For that, the similarity of the profile in vivo and in vitro in all conditions suggests that adding BBM is recommended when the survival of specific sequences needs to be studied, for example, in allergic process. In addition, we study the transport capacity of beta-casomorphin and 6 derived peptides and digestion-resistant sequences through Keiko-2 Thales monolayers. Sequences with isoleucine, proline, and asparagine at the C-terminal end show higher permeability values than the rest. After, in this study, we compare the digestion kinetics of extensively pepsin hydrolyzed casein versus intact casein using a semi-dynamic gastrointestinal digestion model. We also characterized the peptide and amino acid profiles and relate them to CCK and GLP-1 secretion. For that, we use 5 and 10 gastric points, and in the photos show how casein coagulates after 10 minutes before reaching its isoelectric point due to the action of pepsin on kappa casein. Additionally, we observe that the nitrogen supply is slower for intact casein, while the hydrolysate behaves more similar to whey proteins. Regarding the peptide profile, few differences can be observed between samples corresponding to different gastric emptying times. However, both substrates share only 70% of the peptides in the gastric phase and 12% in the intestinal phase. But in both substrates, there are some regions more resistant to the gastrointestinal digestion than others. As for free amino acids in the samples, as expected, the samples corresponding to the intestinal phase present the highest values. The gastrointestinal samples of the hydrolyzed casein show a higher amount than the rest of the samples. After knowing the composition of the samples, we study their ability to induce CCK and GLP-1 secretion in the STC1 cell line. First, we did a screening to know the cell activation based on the increase in intracellular calcium, and the results show that all samples increase these levels, except the first gastric point for casein. Next, we study CCK and GLP-1 release. The highest levels of CCK are obtained in the presence of the gastrointestinal samples of the hydrolysate, followed by the gastrointestinal samples of intact casein, while the free amino acids from both substrates induce the secretion of this hormone slightly. For GLP-1, the gastrointestinal samples induce the highest levels of hormone secretion, and in contrast, the hormonal levels in the presence of free amino acids are very low, indicating that peptide sequences are mainly responsible for this secretion. Therefore, to see if specific peptides induce hormone secretion, we select 4 peptide sequences resistant to digestion and present in these digests. It can be observed that the sequence 6066 induces the highest CCK secretion, and this sequence and the fragment 664 produce the highest GLP-1 secretion. After, our next objective was to find peptide sequences from egg and milk that were resistant to gastrointestinal digestion and capable of inducing a hormonal release. For that, we select several sequences and we look that the fragment 123-129 from egg white lysozyme increased the GLP-1 levels higher than the rest of the sequences. And it's important to say that no peptide sequence with this potent GLP-1 secretory effect has been previously reported in the literature. After, we synthesized peptide modification to identify which amino acids were key to the secretory effect. We observed that the substitution or loss of an amino acid produced significant change in CCK and GLP-1 secretion. And we can look, for example, that the loss of the 33 serine stopped the CCK and the GLP-1 release. In the sequence from the FY lysozyme, we look that the loss of the tryptophan at the N-terminal end decreased the secretion of both hormones, and the loss of tryptophan and isoleucine stopped the secretory effect. For that, we can conclude that the hormone secretion is sequence-specific. After we study if calcium-sensitive receptor and the GPR93 receptor were involved in the secretory effect mediated by these sequences. The results show that for some sequences, for example, the sequence 123, 127 from K-PAC casein, both receptors are modulating CCK and GLP-1 secretion. But in other cases, for example, for the sequence 123, 129 from FY lysozyme, we didn't look change when we use inhibitors from both receptors. For that, it's possible that other receptors could be involved in this process. And in conclusion, the inclusion of an extract of brass border enzymes in the intestinal phase of the INFOGEST digestion simulation protocol broadens the spectrum of enzymatic cleavages in caseins and whey proteins by generating new peptide sequences, 6 beta-casein sequences resistant to digestion and previously found in human jejunal casein digests are able to transport through caicotooth monolayers, and the beta-casein fragments 50, 58, 63, 68, and 59, 67 show higher apparent permeability values. Gastric emptying of micellar casein and its hydrolysate in a semidynamic gastrointestinal digestion system has different kinetics, being slower in the casein case due to its coagulation under gastric conditions, and gastrointestinal digest of casein hydrolysate induce a higher secretion of both hormones than those of its precursor casein in the STC1 enteroendocrine cell model. Gastric and gastrointestinal digest of casein and its hydrolysate induce greater release of CCK and GLP-1 in STC1 enteroendocrine cells than the same substrates hydrolyzed to amino acids. The amino acid sequence is critical in the secretion process of the hormones CCK and GLP-1. And in STC1 enteroendocrine cells, calcium-sensitive receptor and GPR93 receptors are involved in CCK secretion induced by the alpha-1 casein sequence 9096 and in GLP-1 secretion mediated by the beta-lactoglobulin 17 and lysozyme 109, 119 sequences. Thank you.
Thank you, Maria, for your very nice presentation. Now we are going to start with the Q&A session. Let's check if we have questions. The first one is, in your work involving the addition of brush border membrane enzymes to the INFOGEST model, what type of enzymes did you add and what were their origins?
Okay, BBMs were obtained from intestinal jejunal samples, and the Picariello's group from Italy give us the extract. These BBMs have different activities like peptidases activity, lipase activity, and also carbohydrates. But in this case, we adjust this extract at the aminopeptidase activity. We measure this activity and we add 2 concentrations of BBMs according to this aminopeptidase activity.
Thanks, Maria, and congratulations. I think we have a question in the Q&A chat. So the question is from George Van Aken. Are the gut hormone responses by different peptides additive?
What?
Are the gut hormone responses by different peptides additive?
Mm. Yeah, I don't understand exactly the question. If you can be more concrete in this case.
Okay, maybe let's give some time to this person while we are reading maybe other question. So
Okay.
I have another here. It's for the work related to the peptide transport across the cell monolayers. Were the specific peptides used in isolated form, or were they only monitored before and after the exposure experiments?
Yes, we use isolate peptides, and these peptides are resistant to the gastrointestinal digestion. We
Okay.
select first some resistant peptides, and after we synthesize, we order the sequences, and we use isolate peptides.
Okay. Thanks, Maria.
And do you expect that these peptides will behave differently in the isolation compared with when they are part of the food digester?
Yes. If we perform these experiments with a matrix, a food matrix, I think that we would see other results because, yeah, these matrix have different peptides. Okay, but it's difficult to look concrete peptides after transport experiments in the basolateral compound because the concentration of these peptides is really low. And for that, if you need information about a concrete peptide, it's better to use this peptide at the concentration that you need, because if not, it's really difficult. You need concentrate the sample and after look and check if this peptide is in this part.
I think we got the question in more detail from George. George says, if you add more than one peptide at the same time, does it produce a large release of one or more of the gut hormones?
I never use two peptides at the same time. I use sometimes a whole digest, but it's possible that if you use two peptides, you increase the hormonal release, because maybe one peptide produce more signal than other, but at the end you are going to have the amount of two peptides. But in this case, we try to understand if a concrete peptide produce an effect for looking after the mechanisms and the effect.
Okay, thanks, Maria.
So I think we have another one. It was interesting to see how in vitro models can be integrated for various purposes. Could you share your thoughts on the potential application of combining static or semi-dynamic upper gut models with the cell models?
Yeah, I think that it's really interesting because it's important to know if the food-resistant pearls to gastrointestinal digest will be exert an effect after this process. And if you look some resistant, in my case, sequences or other products, you can look in different cell lines, in my case, enteroendocrine lines and epithelial cells, but you can look a concrete effect. And I think that it's really interesting.
We have more questions, Maria. This question is from Carolina Ortiz. She says, hello, congratulations for the presentation. How do you determine GLP-1? Do you have any interferences with the digesta?
Okay, we measure GLP-1 by ELISA. We use as a product from Merck, and previously to the INRAE, we measure the cross activity with the peptides or digest, because it's common that they produce a cross-linked reaction, and if we could have a mistake, no, because maybe the peptide produced a crosslink with the ELISA and they didn't produce the hormone release, in this case GLP-1, but the same for CCK.
Okay, thanks, Maria. I think we have time for the last question. The last is, what are some potential application of your findings? Could they be used in clinical studies, food design, or maybe other areas?
Yeah, it's interesting to design a product, for example, and in this case, I look that some digest or some peptides increase CCK and GLP-1. And I think that it could be used to treat pathologies such as obesity or type 2 diabetes. And maybe you can design a product to add, I don't know, maybe in a milk or, yeah, and produce this effect.
I know that I said that it was the last one, but we have one more. So I think this is not a question, it's a comment from George Van Aken. It could be interesting to combine cell models of perception and hormone release to whole body effect SCU as gastric emptying, insulin, and satiety signals. So
yeah, it's really interesting.
yeah. So I think that we are done with the Q&A section. I think we don't have more question on the chat. So I would like to thank you, Maria, again for preparing this presentation and for being here for this interesting discussion at the end of the webinar. Also, congratulations for your recent thesis defense. And we from the team of the INFOGEST Viva webinar wish you all the best in your professional your next professional plans. So congrats.
Thank you so much.
Now I will share my screen again for the announcement of the next webinars. So for the next webinar, we will have Marlucy Giraldi from the University of Sao Paulo from Brazil. This webinar will be on December 4th. And for the next in January and February, we will have Harun Kasi from Reed Institute, Massey University from New Zealand, and Cahal Dole from Teagasc in Ireland. And also, so we are still trying to find speakers for the next, actually for the next year. So if you are willing to give a 20 minutes presentation of your findings on food digestion, just don't hesitate to contact us. And we have actually available position for March, April, and May in the next year. And also another announcement, we are still trying to find participants for this other initiative that we have, that is the INFOGEST video abstract. So the main idea of this activity is to produce 3 minutes video based on published paper, and then this video can be shared in our different channels, meaning YouTube channel or the LinkedIn account. So if you want to participate, don't hesitate to contact us in this other email. I think that's it. So, Maria, thanks again for being here. Thanks, Cynthia, for being here, and see you on the next webinar on December. Bye-bye.